Hi guys, my name is John. I'm an MD PhD candidate at the University of Utah in the Department of Biochemistry, and I'm in Jared Rutter's lab. And really what we like to study is metabolism and how this is important for human disease. So I'm going to tell you a story about how we used yeast as a model organism in order to understand really complex things in human disease and cancer. So for those of you that might not know what an MD PhD is, I didn't know when I started this program. Really the goal is to get training in medicine, so you can see patients, you can be a regular doctor, but also to get very basic training in science, do research, and understand fundamental questions about human health. So we like to study big diseases, important things like cancer, like heart failure and diabetes, things that really have impacts on human health. And hopefully by better understanding them, maybe we can cure them or at least help people. That's really the goal. So when I started in lab, I was really excited to ask these big questions, to study cancer. But my boss basically told me, I'd be working on this yeast project. So you can imagine my surprise. I was a little bit bummed. And I think this is what most people think of when they think of yeast. They go to the supermarket, they'll buy this bag. It's this powder. You add it to bread, when you're baking something, and it makes the bread rise. And that's really the extent of what people think about. But these are really little microorganisms, and their metabolism is important for things like making bread and brewing beer. And this is what I thought. I thought it was, bread and beer. And when I went into lab, one of the most surprising and satisfying things is when you work with yeast, you actually get the smell of bread and the smell of beer. It's great. But the other really cool thing is that when you go to the supermarket, you're actually buying a strain of yeast that we work with in the lab. It's very similar. So you can do science with something that you get at the grocery store. But really, understanding yeast was this thing that I didn't really care much about when I started. But it was really connecting this up the ladder to a human disease that gave us this context. And I really started to understand how important this was for patients. So I'm going to tell you this story about a little girl. She died before her second birthday. When she was born, she was small. She was only about 6 pounds. She was very weak. She couldn't lift her arms. She couldn't lift her head. She couldn't even really see. She was kind of, stuck in her own little world, and everything was a struggle for her. And you can imagine, this little girl's family, this was their first baby. And the frustration and the heartbreak of your first child having all of these problems that you couldn't do anything about. Doctors tried. They really investigated as much as they could, trying to understand what was going on with this little girl. And they did very careful studies looking at how she got energy from food. And there was this very specific defect, but they couldn't do anything about it because they didn't know the genes to look for. They didn't have an identity for this thing that was so important that caused her disease. And they even wrote about that in their paper that I'll talk about later, that it was just impossible to identify. They couldn't find it because they didn't know what they were looking for. And by using yeast in this project, we started very simply, but we actually gave them this answer. After 10 years, we contacted them and were able to solve this puzzle, piece it together, and really figure this out. So I'm going to tell you a little bit of a story about metabolism. And this is my dog. This is Gideon. He loves to get, excited. He likes to run. He's still kind of a puppy. So, when I come home, he's really excited. He spins around and gets really animated. And that's what most people think of when they think of metabolism. They might think of something like calories or carbs or fat. But metabolism is this really important process that underlies all of biology. And it's important to know that metabolism happens even when you're resting, even when you're laying down. It's not just when you're breathing hard or walking up the stairs and you get tired. It's when you're taking a nap, when you're not doing anything, when you're just sitting and watching this video. Metabolism is still important, and it underlies all processes of biology. But I'm really interested in how food is taken in and how this becomes energy to keep your body running. So at the most basic level, when you eat something, you chew it, you have a good meal, you chew, you swallow, and that's basically the end of what you think about with metabolism and with food. But I'm really interested in what happens in this part. When the food is digested, it gets into your bloodstream, and then it circulates through the blood and is taken up by your cells. But on the structural level, on the basic level, I think we can see evidence of metabolism in other very important areas. And it's shaped a lot of, the animals around us. So like a hummingbird we have here, it has this long beak and this tongue that allows it to access this very long flower that otherwise it wouldn't be able to eat from. The same thing happened with Darwin's finches. So these birds have different sizes of beaks that allow them to eat different types of seeds. And that's really the first step in metabolism, is what you eat and how you get it. And it's driven all of evolution. And it's a very important process that I don't think we think about enough, partly because it works so efficiently. It's just always going in the background. So we don't need to think about it. You can think about it and hold your breath, but you can't think about it and stop metabolism. It's always going. And that's an important point. So like we have here, it's kind of a little bit before lunch. I'm getting kind of hungry, so I'm looking at this. It looks delicious. I could eat any of this on here. But say I take a piece of bread. I chew it, I swallow it, and then it's going to start getting digested. And those nutrients, that food, is going to be broken down and delivered to my cells. And I'm really interested in the part where the nutrients are in the cell and what happens to that to give energy. So once it's in a cell up here, we have these little organelles that are just these fascinating things called mitochondria. And I remember being kind of like shocked and horrified when I was in 7th grade, and I learned that there were these little things inside my cells that gave me energy, and I didn't quite get it. But I just knew they were cool and it was interesting. So it's really fortuitous that I get to study this, and this is my job now. It's kind of cool. But at the most basic level, if you haven't heard what mitochondria is, you might have heard the powerhouse of the cell. But it's so much more than that. It's beautiful. It does so many important things. But at the very basic level, if we take nutrients like carbs or fat, nutrients will be in the cell, and then they have to get into the mitochondria through these channels, these transporters, that really allow these nutrients to go at a very specific rate. Because you don't want to just dump everything on the cell, dump everything on the mitochondria. You want to measure it out very slowly to get the energy as you need it. Because you don't want to just blow through all of your energy if you don't have to. And then once it's in the mitochondria, it goes through different pathways and you get energy from this. So I'm really interested in this process, very focused, but keeping in mind this entire context and how important it is. So the other way I like to think of mitochondria, rather than the powerhouse of the cell, is the mitochondria are like the engine of the car. So you gas up your car. Gas is like food for your car. You kind of fill up the tank and the gas is delivered to the engine, and then the engine runs. But like metabolism, the engine, if you idle it, you keep the engine running, you're not really doing anything. You're still burning gas. And then if you accelerate, if you step on the gas and go really fast, you're going to get lower miles per gallon. And that's like if you're sprinting. You're going to move really fast, but you're going to use a lot of energy to do that. And it's a lot like a car in that way. Another thing that I like to use the car for is the fact that some things on a car haven't really changed very much since the car has been invented. You're always going to have wheels, you're going to have tires, you're going to need to stop, so you have brakes. Those things haven't really changed very much, at least in general principles. The subtleties might change, they might be modified a little bit, but they haven't fundamentally been any different than before. And this really informed biology. I know talking about cars doesn't really seem important for a science talk, but this premise really is important for how we decided to ask a very central question in biology, which is if something has stayed the same, like in a car since it was invented, if it's the exact same almost, it must be important. There must not be many ways that you could get around this. It must have a very important role. So in our system, we use this premise of conservation. So when I say conservation, you might think I'm, conserving a species, or like I want to conserve shark species. Like I'm out there trying to maintain, populations. And it's really a similar process that I'm talking about. It's you want to keep something around. If you want to keep something around and, have it stay the same, it's important. Like, you want to conserve tigers, you want to conserve elephants. Like it's the same premise. But really what we wanted to do is take yeast genes and compare them to human genes and really see what has stayed the same over this gulf of evolution. What has not really been tweaked on too much by random mutation that might, muddle this effect. And this is what we call the conserved overlap here. So this green is our conserved proteins at the mitochondria, and most of these are known. We know a lot about what the mitochondria does. We've been studying it for quite a long time. So they're very basic processes that we've known about. But then there were these things down here, this question mark, these uncharacterized proteins. And that just means we knew nothing about what they did. We had no clue. So we really wanted to study this, and we got lucky and we found some really interesting things. We've studied multiple uncharacterized proteins here and identified their function. But I'm going to tell you about one very specifically that has relevance to that little girl that I told you about before, but also really fits with this car analogy that you need to get gas into the engine. You need to fuel this engine in order to move, in order to survive. So at a very basic level, why would we study yeast to do this? And yeast is a great system. It's very flexible, it's adaptable. You can do a lot of things with yeast that you couldn't do in other systems. And I think the main thing about yeast is we want to take out genes and put genes back in, and other cells might die from this. But yeast are so adaptable that they can tolerate it, so you can really understand what's happening. It's also very fast, it's very cheap, and they smell good, and they make beer. It's fun to work around. So at the basic level, we started very simply with this. We wanted to start with an observation, nothing more, just what happens when we do something, right? The basis of science. So to do this, we use a technique called the spot test, where we'll take different dilutions of yeast at different concentrations, so more concentrated to less concentrated, spot them out on plates of different nutrients, and then just grow them to see if there were any differences under different nutrient conditions. And that can tell us about where this defect might be. So we'll see here, we have a wild type normal strain of yeast like you'd get at the grocery store. No, modifications done to it at all. It grows really healthy, really normally. It has these white colonies, those are the yeast. And then if we knock out a gene, say we take away gene X, these yeast grow much worse. And there's a observation there that in this very specific case, the yeast are growing worse. But that's all the information we had. We knew nothing else about what this gene did. We took it away and they grow differently, but we have no idea what's going on here. So what do we do next? I mean, it's like having a flashlight at the Grand Canyon. You don't really know what you're looking for. So what are you going to do next to really figure this out? And we started, again, very simply. We just ground up these yeast and looked at their different nutrient levels. And that actually gave us a very important clue. So I'm just going to introduce pyruvate. We talked about the mitochondrial pyruvate carrier, but you might not know what pyruvate is. It's basically this breakdown product of these nutrients. So carbohydrates are broken down into smaller pieces, and those smaller pieces are called pyruvate. Pyruvate has to go into the mitochondria, like those transporters I talked about before, in order to make energy. So this was really the step in the pathway that we looked at when we were crushing up these yeast. So when we did this with our gene X deleted, that's now called the MPC, we saw in normal cells, pyruvate levels were pretty normal, they're there, but it's not huge. But then when we take away the MPC, we actually see this increase in pyruvate. And that's very specific. It's like you've blocked this pathway. You've kind of dammed up this channel, and everything upstream of that is building up, but everything downstream slows to a trickle. So like I said, downstream, we have acetyl-CoA. Acetyl-CoA is this immediate step downstream once pyruvate gets into the mitochondria. This was actually reduced when we knocked out the MPC. So we could isolate basically where this block was. If I give this pipe analogy, you have this block here, upstream pyruvate builds up, and then everywhere upstream of that, you also have a buildup, and that's exactly what we saw. And then everywhere downstream, you also see a reduction everywhere. Acetyl-CoA I showed, but everything downstream was reduced. And then you can imagine if you remove this block, you can actually restore the flow, and that's the normal condition. So we had a very specific defect, and we got really lucky here because it was so narrowed in for us that we really only had two possible options. It could either be the metabolism once pyruvate has gotten into the mitochondria, to make acetyl-CoA, the breakdown by pyruvate dehydrogenase, or it could be the ability of pyruvate to get through. So there were only two possibilities. And we started with possibility number one. Very simply, what we did was basically punch holes in the membrane of the mitochondria and just allow the pyruvate to get through. So it didn't need any kind of channel, it didn't need a transporter, and it could get access to this enzyme PDH. So when we look at normal cells, if we look at the activity of this enzyme, we saw that normal cells have good activity. Our MPC deletion, this gene X, has normal activity. It's still there. It can still metabolize perfectly fine if you give it pyruvate. But then this PDH mutant, if we take the enzyme away, we see that it's totally gone, which is a nice control. So we know that when you take the gene away or there's anything wrong with it, you don't get activity, which is just a good control. So the next step was really to see what happened in terms of the transport of pyruvate. So we knew that the metabolism was fine if it could get in, if you could get gas into the engine. But can it get there? Is it like the fuel line is cut or something? So to do this, we used radio-labeled pyruvate. So these little green dots on the end of pyruvate are just radioactivity that we can look at and really tell where pyruvate's going. So we isolated the mitochondria and then just incubated them with pyruvate, either with our gene absent or with the gene present, and then we can compare this and look if pyruvate's getting in or not. And this confirmed exactly what we would expect, that in normal cells, you get pyruvate taken up, the radioactivity increases, and we can see that very well. In the MPC knockouts, we see that you can't get pyruvate in, so it's totally blocked. This channel can't get in. You've cut this gas line from the engine. And then when you put MPC back in after you've taken it away, kind of a take it away and then give it back, you see that this fully restores the ability of pyruvate to get in, suggesting that it's very specific. It's just this channel that's defective in this entire process. The mitochondria are fine. They can metabolize it normally. It's just this one step at that membrane that's blocked. So we have this system now. This is kind of what we've discovered. We have glucose, carbs, going to pyruvate, and then it needs to cross this barrier in the intermitochondrial membrane. So it's kind of allowed through at a very specific rate as you need it. And otherwise, you don't want to just dump everything on the mitochondria. And then once it's inside, pyruvate is broken down by PDH to acetyl-CoA, and then it makes the energy that you need to move. So I know I talked about the yeast stuff and all of our experiments, but I want to get back to that little girl and why this was so important and why I was excited about this entire project. This little girl actually had the observations we made in yeast where I showed the one of two possibilities. Her doctors did the exact same thing. They punched holes in the membrane and showed that once pyruvate could get in, it can be metabolized totally fine. It was just the fact that it couldn't get through this membrane. So they showed the exact same thing that we did in yeast 10 years earlier in this patient, but they didn't know the gene that they were looking for, so they couldn't do anything about it. And this is really, we came in and read this observation and it fit with everything that we'd studied so far. And it was this perfect combination to really make us contact these people and, offer to help, offer our assistance, because they were asking for help in this case report. They didn't know what to do, and they were really frustrated by this. So what I'm showing here is just the amino acid sequence, so the protein of the MPC, across all of these species in a little chunk, just a little frame of this. But going from yeast to fruit fly to zebrafish to human. And you can see in black, they have the residues, the amino acids that are most similar across all of these species. So you can see here, this one that I've indicated by the red star, this arginine, this R. It's a very important amino acid. And the significance of this is basically when I looked in the patient, I solved this kind of three-dimensional, three-layered puzzle. So what we started with was the DNA. We were actually able to get fibroblasts from this patient that they'd saved, so the skin cells from this little girl. They sent them to us, and we were able to look at what was going on in these cells. So while the doctors had done a really thorough metabolic characterization, we kind of suspected that we knew the gene we were looking for, so we wanted to see if that was defective in this little girl. So we started with the DNA, which is the first level of this three-layered puzzle. And then the second layer is the mRNA, and the third layer is the protein that actually has the function. And a defect in one of these or a problem in one of these doesn't necessarily go all the way to the protein level. So it's actually kind of rare that a DNA mutation will have an effect on the protein function. And it might even be totally silent. It might not change the protein at all. So late one night, I was in lab, we'd gotten the data back from the patient, we'd gotten the results. We'd only looked at the very important parts of this gene. And when I was putting them together, it was kind of like, writing down, highlighting different pieces of paper, like very hectic. Like I didn't know quite what I was doing, but I was really excited about it, probably overexcited. So I kind of took longer than I probably needed to. But I basically pieced this together, the fact that the DNA mutation led to an RNA mutation, which led to a protein mutation. And carrying that all the way through in this patient, it actually looked like in this conserved residue, it would have function on the mitochondrial pyruvate carrier. It would have function in terms of getting gas into the engine. And this might be why she had this horrible problem and why she died. And for me as an MD PhD, that was the single most salient part of anything I've done so far in my life, was identifying this human disease that actually caused this little girl to die. And now we know more information about this. And hopefully we can do genetic counseling and screening. And the hope is that her parents got to hear this. I've never heard if they actually have been contacted to understand this. All I heard was they were lost to follow-up. But this was by far the most significant thing I've done, and it was like winning the lottery. If someone offered me the chance to win the lottery or do this experiment, I'd rather do this experiment, even though it was late at night and, no one was around. I was just kind of like, oh, there it is. But it was so cool. So the metabolism of this little girl really mirrored what happens in cancer. It's very similar, and it's the same thing with yeast. So the yeast and the patient looked almost exactly like this phenomenon in cancer. First described here by Otto Warburg, a German scientist who won the Nobel Prize, very eminent biochemist, who did a lot of very basic studies. But what he said was mitochondria in cancer were defective, and that's why you have this buildup of acid and why cancer grows in a different way. And ultimately, he was proven wrong. It was showed that the mitochondria are perfectly fine. It's just that they don't utilize energy in the same way, which is really important for us because, like in the yeast, when I put the MPC back, the mitochondria were totally fine. They just needed this door in order to get the pyruvate in. So this is the very general aspect of this. In cancer, whether there's oxygen or not, cancer cells just want to grow. They want to get as much energy as possible, as quickly as they can, to become the worst cancer they can be, basically. And then pyruvate will get shunted out here into another metabolite called lactate. But also you'll have a buildup upstream of all of these biosynthetic intermediates that just help the cancer make more cells. And what's important about that is it's been thought that if you could kind of relieve this, relieve that block, you could actually start to help cancer patients. So very initially, we wanted to start by just looking at, does the MPC have a role in the Warburg effect? Could this explain part of why the pyruvate is getting shunted and doesn't get into the mitochondria? It totally makes sense, right? You have this inability in these patients in the yeast to get pyruvate in. That would totally explain the Warburg effect. And that's actually exactly what we saw. We saw in colon cancer that the transcript level was significantly reduced. This was in the top 100 most underexpressed genes in this type of cancer. And across many different cancers, it's very underexpressed or even mutated. And then at the protein level, we also wanted to compare, does the normal tissue compared to the cancerous tissue in the same patient have a difference in the MPC level that might explain this? And that's exactly what we saw. So in these two patients, we actually saw the normal tissue has high MPC by protein, and the cancer tissue just adjacent to that has low MPC expression. So, as scientists, if something's low, we want to put it back, right? We want to do the opposite of what's going on and see if it's important in that way. That's exactly what we did. We just decided to re-express or overexpress the MPC in this exact cancer type, colon cancer, and see if that changed anything about how the cancer behaved. So that's basically just relieving this block here. If we go from this case where you can't get fuel to the engine to the case where you allow the engine to function normally, can we bring it back to normal? Can we slow cancer growth? And can we deplete these biosynthetic precursors, these building blocks of the cell, which is what I mean by biosynthetic? That's exactly what we saw. We saw very strikingly, very conclusively, time after time, that when we had the MPC back in these cancer cells, we could change their metabolism, which I'm not showing, and then we could also change their growth. So the spheroid assay that I'm talking about here, these little cancer balls are called spheroids. And what we do is basically grow these, and they'll form little cancer tumors, and we could count these, see how fast they grow, see how big they grow. What we found is when the MPC is back, as you can see really clearly here, not only does the number decrease, so the number of little cancers that forms is lower, the size is lower too. So they're smaller tumors, which is great. I mean, this is phenomenal. The next part we wanted to do, that was an in vitro assay, so kind of artificial. We wanted to do this in mice, get a little bit closer to maybe what a cancer patient would be like. In order to do this, we took those same cells, we injected them into the side of mice with and without MPC, the normal cancer, the cancer where we changed MPC levels. And then we just looked at the tumor growth rate over time to see how fast and how aggressive these tumors were. And like you would expect, since I'm talking about this, it was exactly the same as the spheroids. When you have MPC put back in, there's this slower growth rate, but it also plateaus out here. It just stops growing. So there's something about changing the metabolism that we're continuing to study that actually slows cancer growth. which is awesome. And then you can see the tumor over here, that when you modulate this, when you change metabolism, you actually change the cancer and it behaves more normally. So I just want to conclude by talking about how great yeast is as a model system, how great and important basic science is to lead us to all of these observations. And it was really the way that my boss decided to ask this question. It was the way that we pursued this that really allowed all of these benefits to come from this. We started in a basic system, but the way the question was asked, it was directly translatable to human health. Even if that wasn't the overall goal, that's what we've been able to accomplish here. So basic science is important. Yeast is a great model organism, even for studying something like, metabolic diseases, development, cancer, other things. And then it's really important in how you think about metabolism. It's important to think about how you're getting the gas to the engine. And just a subtle defect in that can have devastating consequences, like I talked about for this family. The other aspect is cancer is very complex, obviously. Cancer is not just one disease, it's many diseases. And there are some cancers with high MPC, some cancers with low MPC, different situations where this will be important. But the really critical thing here is that we've identified this gene, and now we have the ability to study it. We can understand it better. We can do all of these different things in cancer and in heart failure and in diabetes that I set out to do when I started my MD PhD. And that was all because of yeast. But in cancer, understanding this really allows for therapeutic opportunities. So now that we know what the gene is, we know how we can target it, maybe we can activate it, we can do different things with it. If we know that it's low, maybe that's, one weakness of cancer, that if we inhibit something else, we can kill the cancer faster, which is a really exciting potential. And that's what I'm excited in pursuing. So I just want to stop with metabolism is always happening. If nothing else, metabolism is a very important part of biology. It's interesting, and it's not just calories or carbs or diet. It's everything beyond that. So I need to stop by thanking the people, the teams of people that made all of this work possible. They could just as easily be standing up here, but I'm lucky enough to get this opportunity. Dan Bricker was a grad student in Carl Thummel's lab at the University of Utah. He used Drosophila as the first system to knock out the MPC and really understand in an organism what was going on in a larger multicellular system. And he made that first observation that there was this block in pyruvate. Thomas Orsak was a grad student that I overlapped with very briefly that kind of taught me everything I know about yeast in Jared's lab. He started this yeast project and really spearheaded the knockout and characterization and all of this stuff. Eric Taylor was a postdoc, now he has his own lab, and he did a lot of the work on actually the patient fibroblasts and showing when we put the MPC back in, it totally rescues these fibroblasts. Well, we couldn't do anything for this little girl. That was an important thing to show that we could rescue this by just putting the gene back. That was the only thing that was wrong. This next group, our most recent paper are working on the cancer stuff and how the MPC is important. Chris Olson is another MD PhD student with me at Utah in the program. And we really spent a lot of time spitballing ideas and kind of coming up with crazy ideas on how to study this and understand cancer better. So it's been really great to work with him. Jared, obviously. The doctors in France who identified and really thoroughly described this patient. Without their description, we wouldn't have known how important this was for human health. And it was really their call for help and their really basic scientific insight that allowed us to understand this better. Other than that, I have to thank the core facilities at the University of Utah, metabolomics, flow cytometry, imaging. All of these people have been phenomenal in making this work progress at the fast rate that it has. For funding, I'm funded by the Developmental Biology program at the University of Utah on an NIH training grant. My MD PhD program for all of their support and kind of talking me into, melding both of these worlds together and using science to understand medicine and medicine to understand science. It's been a lot of fun. The Nora Eccles Treadwell Foundation for some basic support on how the MPC is important in different diseases. And then Jared's R01. But last, I really have to thank the people that have made this opportunity possible, why I'm getting to talk to you right now. The team at iBiology is absolutely phenomenal. Even though we've only worked together for 2 days, I'm going to go out to San Francisco and I'll have a place to crash, which is great. Shannon, Sarah, Monica, all of them have been just absolutely phenomenal. the Lasker Foundation for making this all possible. Without their generous support, we wouldn't be able to do this. And it's been a great experience, and it's really changed how I'm going to communicate about science. And then especially the Alda Center for communicating science, especially Val and Louisa, making me completely comfortable with everything that was going on here. Even when I was a little bit hesitant, they could see that and kind of be like, no, it'll be okay. Don't worry about it. It's fine. So they've been phenomenal to work with, absolutely great group of people. And then the other young scientists have also been a lot of fun to learn from and talk about these ideas with. So thank you. My email will be on the website if you have any questions about this. I simplified a lot, so if you want to talk about any more complex things, please feel free to contact me. Thank you.